Okay, now plant biotechnology is founded on the principle of cellular totipotency and genetic transformation. In the previous chapter, we had discussed in great detail the basics and some advancements of genetic transformation. But what is cellular totipotency? This is what we will discuss in the slides that's coming ahead. Cellular totipotency is actually the cornerstone for the keystone for plant biotechnology. In 1983, three groups they had described a bacterium that causes crown gall disease in plants, and it's known as Agrobacterium tumefaciens. It is one of the means of introducing new genes into plants, and their genetic transformation, they had been successful in making fertile transgenic plants, for example, introducing a bacterial antibiotic resistant gene into tobacco plants. That was a big breakthrough, which led the scientists to believe that this new technology, the unraveling of Agrobacterium tumefaciens as a gene carrier, as a vector for genetic engineering in plants, it would allow the introduction of important crops, agronomically, agriculturally important genes into species that are susceptible to agrobacterial infections. Not only agrobacterial infections, but it would also pave the way to produce herbicide-resistant plants, as well as frost-resistant plants, those plants that can actually survive in high salinities, because in some parts of the world there is a lot of extra micronutrients in the soil, due to which the soils are having higher level of salinity, more salts are there, and the plants cannot grow. Introducing genes for resistance towards salinity will make them grow in those conditions. Now, what is plant tissue culture? As we said, in plant biotechnology, the two major parts of plant biotechnology is genetic manipulation and totipotency. And totipotency is a way we had discovered, we had started doing things in plant tissue culture. Plant tissue culture is actually it's a technique to grow plant cells or plant tissues or plant organs or plant seeds or any other plants, part of a plant in a sterile environment, in a chemically defined balanced medium. Plant tissue culture actually is the in vitro manipulation of plant cells and tissues, and it is a keystone in the foundation of plant biotechnology. Very, very useful for propagation of plants, not only for the propagation of plants, but also if you want to study the effect of hormones. There are hormones very important for a plant development. For example, auxins, gibberellins, cytokinins, all these are very important hormones. And to study the effect of these hormones, plant tissue culture also play a vital role there. Moreover, when you have genetic manipulation in plants, how will you propagate this? I mean, we do this at cell level. The gene has been introduced into the plant cells, but now this one cell has to become a whole plant so that we can have a genomic transgenic plant. This is only possible, one of the best solution is through plant tissue cultures. And this is possible only when we have a plant tissue or a cell or an organ, and we start from there and we culture it in sterile conditions in a balanced medium, and then they become a whole plant. Every slide is flooded with the word totipotency. What is totipotency? Totipotency is actually, it's an ability of a single cell to divide and produce all differentiated cell in organisms. When we will be studying stem cells later on in the course of biotechnology, we will be discussing totipotency versus pluripotency versus multipotencies. Those are the different potencies an embryonic stem cell can have. But when it comes to plants, there is one word very important that is known as totipotency. Totipotency is the ability of plants. You take any cell, a root cell a leaf cell, a meristematic cell, a clarenchymal cell, you name it. You take this cell, and what you do with it, you grow it. For example, this carrot, there are some cells, you grow it in a grow medium. And now, if you give the proper atmospheric environmental condition and the chemical conditions and the physical condition, this one single cell has the ability to divide and divide and divide and make a mass of loosely cell culture known as callus. Eventually, it will start differentiating into roots and stem, and then you will have a young plant which will eventually become into an adult plant. This is something we can only do with plants. We cannot do it with animals. We cannot do it with any other organisms. But any part of a plant cell, you can culture it and it will differentiate into a complete whole new plant. The history of a plant tissue culture, dates back to 1902 when a Danish botanist, Gottlieb Haberland, he tried to obtain experimental evidence for totipotency. He was working with mesophil tissues from the leaves using NOx solution. He did not succeed in getting this, what we discussed here was a failure for him because of NOx solution. Now, we discussed in the beginning, for plant tissue culture, it is very important to have a balanced growth medium. And if you see the composition of NOx solution, there is calcium nitrate, potassium nitrate, potassium hydrogen phosphate, magnesium sulfate, iron sulfate. These are all micro and macronutrients. This is not enough for the cultures to grow and develop into a whole plant. We need vitamins, we need hormones, and we need a carbon source, which is totally missing here. So this was the reason, but nonetheless, he led us the way. He showed that this can be possible. In 1934, Roger Gottert, a French scientist, had encouraging results with cambial tissues of carrot. Then in the same year, there was another scientist, Professor Philip White. He successfully cultured tomato roots. And then until 1939, Gottert and White, they worked together and they kept culturing and subculturing their cells on fresh media for 6 years. The experiments demonstrated that these cultures could not only be initiated, but they could be maintained for a long period of time. The real breakthrough came in 1962 when a Japanese scientist, Toshio Marasuji, with a scientist named Skoog, they published their recipe for culturing these cells and in vitro conditions. The recipe was of a medium which is known as Murashuji and Skoog medium that is even now used in all plant tissue culture labs for propagation of plants through tissue culture exploiting totipotency. Why is it important to have plant tissue culture? Production of clones. We have a plant that is cloned successfully. How would we propagate it? We need tissue culture for that. Another point which we discussed earlier, quickly, so the speed with which we can produce plants in tissue culture is faster, is relatively more rapid than the conventional way. The absence of seeds, this is interesting. In tissue culture, you do not need any seeds. For example, if you want to plant a mango tree, you need the seed. Until and unless you have a seed, you cannot grow it. But in tissue culture, you don't need a seed. Just grab a plant from the leaf or the root or the flower, anything, and culture it, and you have the whole plant of mango. Then again, the regeneration plant that has been genetically modified. And another good point is you're working in a lab. You have everything under control. So these are the sterile conditions. When the sterile conditions, there are no spores flying of fungi or bacteria, or there are no viruses. And this is why it ensures a disease-free transmission and a disease-free cultivation of the plant.